now I would like to talk about photorespiration, which occurs when the carbon dioxide levels inside a leaf become very low. And this happens on really hot days when a plant is forced to close its stomata to prevent excessive water loss. And if the plant continues to attempt to fix CO2 when its stomata is closed, all of the CO2 will get used up and the O2 ratio, the oxygen ratio in the leaf, will increase relative to the carbon dioxide concentration. So what Rubisco does, not only can it fix carbon dioxide, but it can also fix oxygen. So when the CO2 levels in the leaf go down, Rubisco starts to combine oxygen with RuBP instead of CO2. So what happens here is that when O2 is combined with RuBP, it doesn't lead to the production of useful energy-rich molecules like glucose. So you get these sort of special cellular organelles called peroxisomes that are found near chloroplasts. They break down the products of photorespiration, which aren't necessarily useful. And so the plant uses a lot of energy to break down what it makes, so it's really a negative sort of process. And scientists think that this whole process of photorespiration originated in the early atmosphere, where primitive plants really didn't have that much oxygen. So this is a bad thing, and plants have different mechanisms to do this, to fix this problem. They don't want to use up all of their energy to fix oxygen because that's just inefficient. So plants have actually found a way to stop fixing oxygen with RuBP. They have found a way to become efficient. So upon improving photosynthetic efficiency, some plants have evolved sort of a special add-on feature to C3 photosynthesis. So when carbon dioxide enters the leaf, it's absorbed by the usual photosynthesizing cells, which are the mesophyll cells. And instead of being fixed by Rubisco into PGA, the CO2 combines with PEP to form OAA. And the fixing enzyme that puts these together is PEP carboxylase, and OAA is the first product. It has 4 carbon atoms. That's why it's called C4 photosynthesis. And then this OAA is converted to malate, and then malate is kind of shuttled through the plasmodesmata to specialized cells within the leaf, which are called the bundle sheath cells. And here, malate is converted to pyruvate and carbon dioxide, and the pyruvate then goes back to the mesophyll cells, where 1 ATP is required to convert the pyruvate back to PEP, and then this goes over and over, and the overall effect here is to move CO2 from the mesophyll cells to the bundle sheath cells. So then, the purpose of this is to increase the efficiency of photosynthesis. The bundle sheath cells surround the leaf veins and are themselves surrounded by really densely packed mesophyll cells, and since these bundle sheath cells rarely make any contact with intercellular space, very little oxygen reaches them. So when malate delivers carbon dioxide to a bundle sheath cells, Rubisco begins the Calvin cycle, and because little oxygen is present, Rubisco can fix carbon dioxide without any competition from oxygen. So very little photorespiration takes place, and because of this, photosynthesis is more efficient. And here also, in order for photosynthesis to occur, leaf pores called stomata must be open to allow CO2 to enter, and however, when the stomata are open, water can escape. So the higher rate of photosynthesis among C4 plants allow them to reduce the time that the stomata are open, thereby they're reducing their water loss. And so C4 plants are found in really hot, dry climates where they have an adaptive advantage. And so some examples of C4 plants include sugarcane, corn, and crabgrass. So plants actually have another type of add-on feature to C3 photosynthesis, and that's called CAM, and the spelling out of that is crassulacean acid metabolism. So it's kind of a lot like C4 photosynthesis, but some things are different. So I'm just going to say these differences, that PEP carboxylase, it still fixes CO2 to OAA, but instead of malate, that OAA is converted to malic acid. And that malic acid is, instead of being shuttled to the moved out of the bundle sheath cells, it's moved into the vacuole of the cell, and at night the stomata are open, PEP carboxylase is active, and malic acid accumulates inside the cell's vacuole. During the day, the stomata are closed, which is the reverse of other plants, and at this time, malic acid is shuttled out of the vacuole and converted back to OAA, which actually requires 1 ATP to ADP, and it releases carbon dioxide. So now that CO2 is fixed by Rubisco, and the Calvin cycle can proceed. So the advantage of CAM photosynthesis is that it can go through the day when the stomata are closed, and you really don't lose that much water, which is great. And as a result, CAM provides an adaptation for plants that grow in sort of hot, dry environments with cool nights, so basically deserts. And this, that name, that really weird name, crassulation acid metabolism, comes from the early discovery of cam in succulent plants. It's kind of like pineapples, and also the main thing that you can use this to remember is cactus. Cactus have cam. Okay, so we are done. We basically talked about everything you need to know about photosynthesis. We discussed the light reactions, the dark reactions, cyclic and non-cyclic photophosphorylation. We talked about the Calvin cycle as well as chemiosmosis, and we also briefly discussed the structure of chloroplasts. And we also talked about photorespiration and different fixing mechanisms such as C4 and CAM photosynthesis. I'm Saba, and I'm out for today. That's all, folks.